Hello, in this video we will understand about the SNP genotyping. So what is this? The single nucleotide polymorphism. So in this way the single nucleotide polymorphism means the single nucleotide variation within the population which that will change the variation in the color in the same population which that will lead to changes the phenotype. So remember that every individual within the population contains cell and cell contain DNA and DNA contain a gene, but the same gene containing one nucleotide changes. This one nucleotide changes can be a adenine instead of the thymine or thymine instead of the adenine, which that will lead to change the whole color or whole pattern. So the human genome carries at least 10 million nucleotides position that vary in at least one of 100 individual in a population. So in this way, at least one of the 100 individual, it varies. So the identification of these single nucleotide polymorphism SNPs is important tool for identification of genetic loci linked to complex disorders. So remember that basically the nucleotide position that vary in at least one of 100 individual in a population which that will lead to changes, and we can identify and understand the disorders and disease. So this is the chromosome which that contain alleles. Alleles are present in the chromatids, the sister chromatids of the same chromosome, and the locus is where the gene is present, and the centromere is the center part. So the alleles contain a gene basically, and this gene dominant or recessive on the basis of situation, the dominancy, heterozygous or homozygous condition. So this is the a population of the human contain different individual contain different cell. But remember the all individual gene is same, but one individual cannot match only one nucleotide as a single nucleotide polymorphism. In this way, other also change, and that is why the changes which that will lead to appearance will be changed. The appearance changes due to the one nucleotide. One nucleotide change mean the, for example, the cytosine instead of adenine, which that will lead to whole polypeptide synthesis from the messenger RNA into the translation into the production of protein will be different as compared to the others. So let's begin to understand the identification SNP genotyping. So this is a cell which that basically is containing here is the adenine, cytosine, guanine and thymine, and the one labeled here. So basically here is the blue color is labeling for the cytosine only. So the reverse transcriptase enzyme also adding, and the several segment of the RNA which that will lead to reverse transcription in the PCR, and extension will lead to hybridization to produce different segments which that on the basis of the terminal region contain a cytosine containing the blue color fluorophore if the present and the termination, but remember this some segment will not but some segment have. In this way we can calculate the SNP1, SNP2 and SNP3 genotyping. SNP1 means the adenine only, so no color, and the guanine and cytosine basically will produce color, and on the other hand the cytosine will also produce color because the cytosine. So in this way the SNP genotyping identification through a fluorophore and by the detector, and in this way we can change we can detect through color On the other hand, here is a different method in which the labeled nucleotides adenine, guanine, cytosine, thymine, dideoxynucleotide, uh, uh, mm, nicotinamide triphosphate, which that will lead to label the different colors. For example, four color, and this four color blue and orange, white, and you can understand this is the green. And we can add also PCR products and DNA polymerase for synthesis and increase the amplification of the segments. When increase the segment increase in this way, for example, when the terminal region will be T if the thymine, so the specific probe will be attached for the production of color, and this fluorophore will produce color after the termination of the each segment. So the SNP1 genotyping contain adenine. So that is why the adenine is the terminal. So in this way, the opposite side will be thymine, and in this way the opposite thymine attached will lead to produce color, and this detection you can understand in this manner, and this produce the color adenine, which that is SNP1 genotyping and SNP2 genotyping and SNP3 genotyping. These are the different type of genotyping when we will add the basically the production of the on the basis of nucleotide variation. So product first in the first time in the primer extension will lead to in solution if in this way SNP1 genotyping is the green, SNP3 genotyping is the orange, SNP2 is the mix. So in this way we have gained the products. In this way we will again amplification and capture that color for identification, and we can understand where is the variation. So in this way the different segment again produce, so the SNP1, SNP2 and SNP3, which that contain same color, and in this way we can identify where is the color is changes. If the SNP2 genotyping color will not change and different as compared to the other before the after the extension, it means we can say that the SNP genotyping, the single nucleotide polymorphism are present, which that will lead to change the individual. So the comparison within the population, the here you can see this is the changes in one nucleotide. For example, the cytosine and guanine should be equal, but here is the difference. So that is why these changes will lead to synthesis of that protein will different the nucleotide polypeptides. So that is why the different polypeptide will produce a different type of protein, and their folding and their different type will produce a different colors and different things. So that is why in this manner you can understand the behavior, color of animal, shape of animal as well as plant, which that is different due to the one nucleotide variation within the population. That is why it is known as a single nucleotide polymorphism. And remember, the SNP genotyping is a single nucleotide polymorphism. Due to the one nucleotide variation will lead to whole change the individual within the population. Thanks for watching.